Hello, I am Mark Stevens from the Shropshire Lichen Informal Group. With the aid of Lottie Stevens, we have prepared this short video to demonstrate the use of Mark Powell's ink vinegar staining method for staining lichen-related structures. The results of the ink vinegar staining method shown here can be used to aid the study of the anatomy of the intact lichen, for example the apothecia, or to examine isolated structures such as the spores or paraphyses. This video will take us from the point at which thin sections have been cut from the lichen to the point at which slides are ready for detailed microscopic study. It will only show the preparation of a squash for spores. Similar but slightly different techniques would be used for the preparation of other samples, for example, a spore squash in water or a cross-section of an apothecia to study its anatomy. Apart from the dissecting microscope stage, from where you have cut the sections of the lichen, this picture shows the kit required for the process. This comprises the following: glass slides, cover slips, pencil with a rubber end, ink, Parker's blue-black ink is recommended, acetic acid, white vinegar, a bottle of K, that is potassium hydroxide or sodium hydroxide, approximately 10% solution. This is the bottle you will be using for chemical tests on lichens from your kit. Tea lights and matches, fine forceps, cocktail sticks, a tray to put instruments in, tissue paper, a plastic pipette for water, and nail varnish. Label most of the pieces of equipment. For example, in this picture you can see that I've labelled the water, the dropper bottle of K, the acetic acid, the pipettes, and so on. It is important to label each glass slide with the key details of the sample that you are taking. It is quite easy to get your slides mixed up if you are making more than one type of preparation in the same session. It is also worth selecting out one glass cover slip and move it to a convenient place so it is easy to pick up during the process. In this case, I have put it in a separate chamber in the cover slip box. Arrange the equipment for optimal use. This photo shows the kit arranged in a way that I find useful. It might take you a few goes to work out the best places to put things. It is quite easy to end up in a bit of a mess, particularly if you combine the staining task with the previous task of cutting sections. As a warning note, you've probably noticed that I'm left-handed, and if you are right-handed, you will probably arrange the kit differently. Application of K onto the slide. Apply just one drop of K onto the labelled glass slide. Application of ink. The next thing to do is to apply a drop of ink to the K. I use a cocktail stick, and you will see that this may take a couple of goes to obtain a good ink drop. Transfer of slices of lichen to the ink. Transfer the previously prepared slices of lichen to the inky drop. Wetting the forceps tip aids picking up the lichen slices. Transfer the material using the ultrafine forceps under direct vision, looking down the dissecting microscope. Transfer as many lichen fragments to the ink as you wish. Heating the slide. Light the tea light and warm the inked area of the slide over the flame. Hold the slide approximately 2 to 3 centimeters above the flame. The aim is to get the stain hot but not to boil it dry. Good heating facilitates good staining. Applying the cover slip. Move the previously prepared single cover slip from the cover slip box with the ultrafine forceps and lower it gently onto the inked area. Squashing the spores from the apothecia. Using the rubber end of the pencil, tap this vigorously on the cover slip with the slide supported on a firm surface to obtain good separation of spores from the apothecia. Clearing the ink. Place some tissue on one side of the cover slip and then apply some acetic acid to the other side of the cover slip between the cover slip and the slide as best you can. The acetic acid will sweep through the preparation between the cover slip and the slide, clearing the excess ink. This will be aided by the capillary action of the tissue. You may have to reapply the acetic acid until the preparation goes clear. Drying the slide, removing the excess liquid. Hold the slide in your hand and wipe the slide dry along the lower surface and around the cover slip edge on the top surface of the slide with the tissue. Be careful not to wipe the cover slip off when you do this, which is quite easy to do. It is quite easy to knock the cover slip a small amount, but as long as it's not knocked off, just reposition it. Check the results. Take your slide to the compound microscope and check your work. Check to see if the structures of interest, in this case spores, are suitable for further analysis. Sealing the slide. The ink vinegar preparations are not permanent and will dry out. So assuming the preparation is sufficient for further examination, to decrease the speed of drying, seal the join between the cover slip and the slide with nail varnish. In this case, red nail varnish, but other colours will work fine. Storing the slides. The slides will be perfectly fine for examination over at least 24 hours and probably more, but will eventually dry out. That completes the video of the ink vinegar method of staining for lichen structures. I do hope you enjoyed watching.